My name is Nick. I've been working in Beth Mormino's lab for the past few months for the NST, and I'll tell you today about regional tau asymmetry in PET imaging, and I have no disclosures to report. Sorry, this isn't responding here. Okay, well, I'll just go off this. So the purpose of today's talk, hopefully I'll convince you of the importance of tau PET imaging, particularly in Alzheimer's disease and asymmetry. I'll explain the little that we know about asymmetry in tau PET and then provide some of my data analysis on the subject, as well as potentially how it may relate to clinical phenotype and provide some summary points and future directions. So we're all obviously familiar with Alzheimer's disease. It is, of course, the most common cause of cognitive impairment in ages 70 through 90. And since the mid-1800s, we've known that amyloid plaques have been composed of beta amyloid protein. If you fast forward about 20 years from that, we've been able to image this amyloid real time in humans through Pittsburgh compound B in amyloid PET. And with all the research in amyloid, fast forward 20 years, lecanemab has been put forth as one of the disease-modifying therapies that's anti-amyloid. Now, despite all of this kind of research in amyloid, it is only 1/3 of the ATN classification of Alzheimer's disease. Tau and neurodegeneration are the other two parts. And I think tau, for a number of reasons, has particularly interested me in terms of its PET imaging features. So tau, of course, is a protein that has many cellular functions. It's particularly important in stabilizing microtubules, and it's known to be regulated through phosphorylation. Now, when tau is hyperphosphorylated, it tends to aggregate, and that can occur in many different forms in many different diseases. We have PIC bodies, the GGTs that I talked about in my grand rounds last year, and of course, neurofibrillary tangles in Alzheimer's disease. And these neurofibrillary tangles tend to progress in a very classical stereotyped fashion that was first demonstrated on autopsy studies by Brock and Brock in the early 1990s with a transentorhinal through mesial temporal through isocortical spread. However, there are many atypical kind of signatures that don't fit with this, and we've really only discovered this fairly recently, as we've only really had a good tau PET tracer for the past 10 years, as fluortaucipir was first developed and used in humans in 2013. There are a lot of advantages to it, even though there have been later tracers. It's actually fairly specific for the neurofibrillary tangles versus other tauopathies compared to some of the other newer tracers, and it's actually FDA approved. As recently as 2020, although I'm guessing most people in the audience have really never seen this ever used before. However, there are a number of advantages to it versus actual amyloid PET imaging. So spatially, tau PET shows concordance with Brock staging in kind of like autopsy sections. There's concordance in the imaging pattern with hypometabolism and FDG PET. There's concordance with areas of atrophy on structural imaging, and then with worse performance on cognitive testing and prediction of cognitive decline. So for such a powerful imaging modality, I think there's a lot of questions currently. Can it be used for diagnosis and staging of Alzheimer's as well, but also identifying atypical forms of the disease that don't fit with the Brock staging model? And I was particularly interested in this and the question of asymmetry, which we've really only discovered fairly recently. And work from our own lab, Dr. Christina Young has identified even in asymptomatic, cognitively unimpaired older adults, up to 1 in 11 or so have that are amyloid positive have an asymmetrical tau PET signature on their brains. But we don't really know what this means because we don't have great longitudinal data from these individuals. Groups using machine learning to look at tau PET and kind of classifying images kind of based on longitudinal kind of courses have kind of concluded that laterality increases with the disease state. And then a study from last year looking at the left side of the brain versus the right side of the brain showed that tau PET asymmetry was associated with an earlier onset of disease, as well as increased total and phospho tau and increased kind of decline over time. So all kind of bad things from asymmetry. So I was really interested, is asymmetry bad? Does it progress with disease? Does it get better? And like, what does it actually mean for a tau PET? And so to do this, I've looked at a variety of imaging kind of platforms. I'll tell you about 2 different data sets today, the ADNI or Alzheimer's Disease Neuroimaging Initiative and the A4 study, which is largely asymptomatic adults. And I've defined 6 regions of interest for what I'll be presenting today at least, which are the frontal and occipital lobes, as well as lateral and medial temporal and parietal lobes. The index I'll share today that I've used for measuring laterality is a laterality index. It's a fairly straightforward concept. You essentially take the left-sided signal, you subtract the right-sided signal, and then you divide that by the absolute value of both. So a very positive result being a very left-sided signal, a very negative result being a very right-sided signal. And to measure the total amount of laterality, you can kind of take the absolute value of this. And then for some subjects, I've looked at a control group defined as unimpaired individuals who are young for our subfield, which is less than 65, who are amyloid and tau negative on PET scans. So one of the first things I did was just map the total amount of laterality index or laterality by total tau. And there are a lot of small graphs here, but for these 6 different regions, you can see laterality index is on the x-axis, and more positive values would be to the right, meaning more left-sided, and more kind of right-sided signal would be to the left or more negative. And you can see there's this large cluster that's yellow kind of at the bottom of all of these different regions. These are individuals I've coded as yellow as unimpaired. And all these regions look pretty similar, but I noticed there was a little bit more spread in some of the lateral regions, including lateral parietal. So for today, I'll really focus on this region. And just to kind of enlarge this graph more, you can kind of see this spread. And then some of the red and green individuals who have MCI and dementia, you can see have total higher tau. They're higher on the y-axis, and they have a little bit more laterality too. And so it looks pretty symmetrical. There's a few more individuals on the right of the graph, so being a little bit more left-sided. But to simplify things, I'm going to show you absolute values of laterality. So being more to the right would be a higher overall laterality index. And you can see there's a spread, and there's kind of almost 2 distinct groups here besides that kind of group at the bottom left of the graph. So there's a lot of individuals with a lot of total tau and not a lot of laterality. And if you note, they almost all have dementia, they're all green, and they all have positive filled-in circles, which means they're amyloid positive. Now there's individuals and if you look at their actual images, they all have amyloid all over their brain, and their tau signal is fairly symmetric. Now, if you look at individuals on kind of this bottom right part of the graph who have high laterality and moderate amounts of tau, they tend not to have a ton of amyloid. A lot of that is kind of non-specific signal, and their tau signal is like very asymmetrical. A lot of these individuals are also amyloid negative, which is a little confusing because traditionally we think you need amyloid before tau in the Alzheimer's disease kind of like course, but more on that later. So I was really interested in kind of looking at these individuals and figuring out, are they gaining laterality? Are they losing laterality with time? And then what is amyloid kind of, how is amyloid affecting them? How is tau affecting them as well? So to look at kind of what's happening over time, these are individual scans of individuals, and some of them we actually have multiple scans. Again, this is a fairly new technology, so we don't have like great data from like 10 or 20 years from individual patients, but I connected individual scans just with simple arrows on these graphs to show you kind of what's happening. And you can see the y-axis, they're almost always slightly increasing, which makes sense because the y-axis is total tau, you're gaining total tau, but some gain laterality and some lose laterality over time. So different individuals seem to kind of act differently, but there's often kind of a similar pattern when there's multiple time points. And when I've looked at a lot of individuals with this kind of approach, I've noticed that individuals who tend to have less total tau tend to kind of gain laterality. And there seems to be this inflection point where after a certain amount of tau, you lose laterality and you kind of revert back to a more symmetrical state. When I've looked at individuals in this group who have a ton of tau but don't have a ton of laterality, there's really no clear trend. They kind of go all over the place. So when I kind of map all these individual scans and connect the dots, meaning like which are kind of the same individual mapped over time, again, you can see kind of at the bottom middle of the graph where there's not a lot of laterality, some individuals just kind of go straight up on the y-axis. They just kind of gain tau, but a lot of individuals fan out to the side. They're gaining laterality, but that seems to kind of stop at a certain point. And then if anything, individuals kind of revert back to the mean, or they just kind of progress upward as well. And so figuring out what distinguishes these different people, I think is really interesting, especially if this may have clinical relevance. The other thing I was interested in is, again, this group on the far right, I mentioned doesn't tend to have a ton of amyloid. And when I filter out individuals with less than 5 standard deviations of total tau, so kind of getting rid of that big clump at the bottom without a ton of tau, you know that there's a pretty big difference in the amyloid positive and negative individuals in terms of laterality. The amyloid negative individuals have about twice as much laterality. However, you also note that total tau seems to increase as laterality, or total tau seems to kind of be higher for individuals with less laterality. So, to really get at this question kind of better, I needed to do a linear regression. So when you look at laterality, amyloid, and total tau kind of all in the same model, and you filter individuals at that kind of inflection point I talked about, where they reach a certain amount of laterality, then they kind of revert back to more symmetrical patterns. Interestingly, total tau isn't associated with laterality, even though it kind of appears like it on the graph. And there are significant associations with laterality and amyloid. So briefly, laterality seems to increase with age, but with increased amyloid, laterality seems to decrease as well, which is a little counterintuitive. But my conclusion is that as you're aging, laterality is increasing, but it seems like amyloid has a strong effect, which might potentially decrease laterality as well. I heard Dr. Porter's timer, so really briefly, I'll just mention there's a few clinical associations with this. It seems like laterality is kind of worse for your MOCA scores, at least for clinically even unimpaired patients, but potentially even for MCI and dementia, that's more of a trend. But certainly when you kind of get higher into dementia stages, laterality is kind of worse for many cognitive domains, and left-sided laterality, as you might expect, would be worse for language in most individuals as well. And so really briefly, again, with this model, it seems that in many individuals, they kind of gain laterality with time, and then there's a certain state where you maximize laterality and you revert back. And it seems like potentially increased amyloid might be mediating that, although I certainly need to do kind of further work on this. And then laterality also might influence clinical phenotype as well. And so further questions will relate to looking at other cohorts, to looking at like clustering properties of this, and then applying machine learning techniques as well.
Fantastic. All this work was done in Aaron Gibson's lab, and we'll go over everything that I've done. So first, I have no financial disclosures. I wanted to start with a brief overview of what we're going to be talking about today. So we're going to go through a very rapid overview of the circadian machinery, as well as what happens when these rhythms are perturbed. We'll talk specifically about circadian rhythms in the oligodendrocyte lineage, and then my ongoing experiments in the lab. So first, I wanted to briefly talk about time in biological processes. Humans tend to think of time as a uniquely human concept, but it's actually inherent in all living organisms, whether it's on the minutes to hours level, to days, all the way up to years and decades. And I've included a picture here of the cicada as an example of that. We'll be talking mostly about circadian rhythms today. So that's a rhythm with a 24-hour periodicity. It's called circadian from the Latin circa diem, or about one day. And there are intrinsic molecular mechanisms that establish these rhythms in cells. They're very cleverly called clocks. There's two important features to know about these clocks. One is that they are self-sustained, and the other is that they are actually influenced by external factors called zeitgebers, or time givers. So we're going to do an overview of the circadian system in 2 minutes or less. We'll start at the molecular level. Clock and BMAL1 are two transcription factors that, when they heterodimerize, translocate to the nucleus and transcribe clock-controlled genes, the products of which have a negative feedback loop that help establish this 24-hour rhythm. At the organism level, we actually have a circadian pacemaker in the brain at the suprachiasmatic nucleus, and this helps regulate behavior as well as homeostasis on the 24-hour cycle. Now, the suprachiasmatic nucleus, as well as other cells in the body, have this self-sustained circadian rhythm, but they can be influenced by external factors, these zeitgebers, the most notable of which is light. So light can be transmitted through the retinohypothalamic pathway directly to the SCN, and that impacts how the body can adjust the circadian rhythm. So for instance, with jet lag or with things such as shift work. So this is how the human circadian system works. What happens when we mess it up? So the most common example of this is shift work, and I've included a few photos here of some of the participants in the room during their night shift. And it's been well studied that you can have adverse health outcomes due to prolonged night shift. And I want to just emphasize, this isn't just from perturbed sleep. This is actually misalignment of both metabolism as well as gene transcription. But sleep does play a role as well. So shift work can be associated with things like increased rates of cancer, stroke, MI, metabolic syndrome. And in a special population, in pregnancy, you can see an association of shift work with preterm labor, hypertensive disorders of pregnancy, as well as small for gestational age birth weight. Now, this is interesting. This might have social implications too. So some of our collaborators at WashU were doing a prospective study of pregnant socially disadvantaged women, and they found that in these women, when they had an interdaily sleep deviation with increased rates of this, they actually saw an association with MRI findings in the infants 1 week after birth. So they could see a reduction in cortical gray matter, smaller white matter volumes, and reduced cortical surface area. So that's in humans. What about in mice, which is where we study most of circadian rhythms? So in mice, we can see transplacental transmission of the circadian rhythm, circadian entrainment, and you can see the clock genes expressed as early as day 10. We do see that the fetal SCN, suprachiasmatic nucleus, shows rhythmicity about 3 days prior to birth. And this entrainment is hypothesized to be both through humoral signals. So melatonin is the one that is most widely studied, but also glucocorticoids and dopamine, as well as other signals, things such as glucose utilization and activity. And they have looked at what happens when you disrupt this system and the effects on mice. From a neurobehavioral perspective, there was a study looking at an 8-hour phase delay in pregnant mice and what the effects were on the pups. And they actually saw increased stress behaviors in the offspring, as well as altered rhythmicity of the clock genes. Most interesting, these stress behaviors actually persisted up to a second generation after they did this experiment. All right. So just to summarize real quick, we've talked about the circadian system, about how we have these cellular clocks that are both self-sustained but can also be influenced by external factors. We've also discussed how when you perturb this system, such as with shift work, you can see human disease. And we also know that these signals for circadian entrainment can be transmitted across the placenta. What we don't yet know is what happens when you disrupt the circadian system in a developing brain, and how does that affect the brain overall? So this is a really complex question. I'm not going to answer it in my 4 months of research, and I'm not going to give you the answer in this 13-minute presentation. But I wanted to emphasize that we have multiple layers of circadian regulation, and we also have the huge complexity of the developing brain. So to better focus this question, I actually zeroed in on a specific cell lineage, and I chose the oligodendrocyte lineage. There's a few reasons for this. The first is that we know there's a role in neurodevelopment. Over half the CNS is myelinated white matter, and we know that oligodendrocytes are responsible for this myelination, and they are derived from oligodendrocyte precursor cells, or OPCs. And most importantly for this project, we know that in OPCs and oligodendrocytes, the circadian machinery is present and important for function. And how do we know this? Because we've knocked it down. So BMAL1, if you think back to several slides ago, we talked about how there's a heterodimerization of transcription factors. One of these is BMAL1. We've done a cell-specific knockdown of BMAL1 in mice and then characterized this phenotype. And what we found is that BMAL1 knockdown in OPCs results in decreased OPC proliferation, decreased density in OPCs, as well as changes to the morphology. Okay, so that's precursor cells. What about the actual oligodendrocytes? We see changes persist in that too. There's still decreased density in the corpus callosum, cortex, locus coeruleus, and importantly, we also see structural differences. There's thinner myelination in these animals who've had the circadian clock knocked down in the oligodendrocyte precursor cells. All right, what about function? Do these animals act any differently? Well, we have looked at sleep in these animals, and what we've seen is that even between the wild type and knockdown animals, total sleep time is very similar, but sleep fragmentation is increased. These animals have a more difficult time sustaining wake periods, and will have sleep during that. So looking at these animals, we've shown that from a genetic targeting perspective, if you knock down the circadian clock in OPCs in the oligodendrocyte lineage, you can see effects. But what about environmental disruption of the circadian clock? And that's the project that I've been working on. So what we've done is created a model to cause circadian disruption without impacting total sleep time. And how we're doing that is with chronic jet lag. So we take pregnant mice, and at embryonic day 7, they either maintain in their control setting, which is a 12-hour light-dark cycle, which is what you see in most animal houses. For the experimental group, we subject them to chronic jet lag, which is every other day, they have a 6-hour phase advance of their light-dark cycle. That's the equivalent of if you flew from Chicago to London every other day. Then at birth, we revert the animals back to the control cycle and then monitor weight. And at 21 days of life, after developmental myelination is mostly complete, we harvest the animals and then look at histology as well as electron microscopy to get a better sense of what we're looking at. So I want to emphasize, similar to Gabby, that these are very preliminary results, and there are ongoing experiments to both replicate this and to expand on this work. But what we've seen is that mice who are subjected to chronic jet lag in utero have increased proliferation of oligodendrocyte precursor cells, specifically in the corpus callosum where we looked, yet there's no significant difference in their cell density. So increased proliferation, yet similar cell numbers. What does this mean? Well, there's a few different things that it could mean. The first is we need to replicate this. The second is, could this be differences in cell fate? Are we seeing increased cell death to explain these same cell numbers? Is there a difference in differentiation? Or are we seeing this increased proliferation because we're shortening the circadian cycle by causing the animals to be chronically jet lagged? So by causing a continual phase advance, it actually tends to shorten the circadian cycle. We know that the circadian cycle is intimately entwined with cell replication, and that may perhaps explain some of these results. All that to say, the initial results that I showed, it's an N of 4 and an N of 3, so we need to do this a few more times to get a sense of if this is true and what exactly it means. Additional work that we're doing to kind of parse this out is we have the phase advance model that might be shortening the circadian rhythm. We're also looking at a phase delay, pushing things out, and then we have an irregular light-dark cycle model. And this is really interesting. This is actually derived from human data from our collaborators. And it's from patients who report their sleep onset as well as their wake time. And you still get the total sleep time, but it's interrupted and very fragmented. So we're using this as another model for circadian misalignment. Additionally, we still have the chronic jet lag project, chronic jet lag model that I was talking about. We still have further histology and electron microscopy studies to look at both cell types, cell numbers, and myelination in these mice that we're stressing.